Okay, we're going to get started in just a minute. If everyone can start moving in and take your seats, please. Okay, so thank you everyone for coming back in and joining us again. Just a couple quick announcements before we introduce the next session. So we know that it's a little bit chilly in the room. We have alerted the hotel, so we are trying to bring the temperature up a little bit in here. We appreciate your patience as we work on that. Again, a reminder that if you need to double check the agenda for any reason, the barcode on your name tags will provide any information that you need. And one other just quick reminder, or I can't think of the word right now, but we did add mic podium stands for questions during the session just to make it a little bit easier. So we invite you, if you're able to, once we open for questions in our panels, please feel welcome to stand up and go to one of the podium mics on either side of the room. If you would prefer to stay in your seat, please raise your hand and we will run a mic to you. But we do have the two stands to make it a little bit easier for questions. So with that, I am going to hand it over to Dr. Collin Hovinga to introduce our next session.
Thank you. Wow, you can actually hear me, which is good. I hear myself, which is also good because usually my voice doesn't carry, so I'm thankful. I want to thank the panelists for joining me today. You can't just hear the microphone. Can you all hear me? Is it coming through? Which do you want? Lavalier? Okay. Okay, can you hear me? Okay, got it. Okay, so you just started glitch myself. Mary, would you go ahead and start to introduce yourself? I'm going to have panelists work from far to near, introduce themselves and why they're here. So thank you.
I'm Mary Short, and I'm here today representing the real-world data and real-world evidence subgroup of the Cell and Gene Therapy Working Group of the International Neonatal Consortium.
Yeah.
I'm Hidefumi Nakamura. I'm from Japan. I'm a director for clinical research and development at National Center for Child Health and Development. I have some experience at the PMDA, a former body of PMDA, so I know regulatory science. Now, by training, I'm a pediatrician and pediatric pharmacologist, and I'm in charge of a network called Japan Pediatric Society Drug Development Network. That's a network of advisors from different subspecialties from Japan associated with Japan Pediatric Society, just like AAP here. And well, I'm myself not involved in gene therapy, but I have very close connection with the people involved in gene therapy. So I'm trying to do the best to introduce about Japan.
Right. And hi, everyone. It's a great opportunity to be here. Appreciate the invitation. I'm Pat Gonzalez. I'm the head of clinical science at Solid Biosciences. My background is in muscle biology and physiology. And so I'm really representing the clinical development perspective here as our pipeline focuses on gene therapies using the AAV-based platform for rare neuromuscular and cardiac indications.
Good morning, everyone, and thanks for the invitation as well. I'm Najat Bouchkouj. I'm a pediatric hematologist oncologist and the Associate Director for Pediatrics in the Office of Therapeutic Products in the FDA. I lead initiatives that are scientific, regulatory, and policy focused in terms of developing cell and gene therapy for pediatric patients.
Hi, good morning. It's great to be here with you. I'm Keely Milligan. I'm from Novartis, and I'm here representing the pediatric point of view. I work in the Pediatric Center of Excellence and also closely collaborated with the Zolgensma team.
Okay, thank you. We're going to start the conversation with sort of setting the table. And Najat, would you give us sort of the FDA considerations and walk us through how gene therapies are approved and particularly laying on the pediatric context?
Sure, I'm happy to provide that. In terms of the approval for cell and gene therapies, as you all probably are aware, it starts from the interactions from at the pre-IND stage, and then followed by the pre-IND, then the IND, then the BLA and marketing application. And throughout the life cycle of cell and gene therapy product development, there are several interactions that could happen with the FDA to help guide sponsors, academicians who are conducting the trials, and trying to advance these therapies forwards for patients. At the Interact meetings, which replace the pre-IND meetings, are non-binding, very informal, where sponsors can receive a lot of feedback in terms of like just overall general product developments, and then move on to the pre-IND, and then when the IND comes in, that's when more meetings occur between the FDA and the sponsors. I want to emphasize that we always encourage and ask sponsors to involve the patient's voice from the early stage of product development. Don't wait until the IND is submitted or the application is about to be submitted for marketing authorization. The conversations with the FDA should really include patients from the early stages so that their voice is really heard. As we heard earlier, the patients are the end product who are going to receive those therapies, and it's really important for them to be sitting at the table when the clinical study protocols are being designed, to really know like what's important to them, what's really going to be changing their life as they're receiving end of the therapies. Obviously, there are certain regulations that the FDA work with. There are legislations that are in place to either mandate and require studies in pediatrics, or that incentivize sponsors to conduct the studies in pediatrics. There's either the PREA or the Pediatric Research Equity Act, which was enacted in 2003, and it mandates and requires sponsors who are developing therapies for adults to also conduct the trials in pediatric patients. And then there's the BPCA, which is the Biopharmaceutical Children's Act, which is an incentive program that incentivizes sponsors to conduct the trials in pediatrics. So there are, several avenues where the regulators can really interact with the sponsors, with the patients, scientists, to move these therapies forward.
To add to that, are there any like specific guidances that you find yourself, recommending people be familiar with as they're prepping and trying to understand the considerations that they need to do to design and conduct gene therapy trials?
So there are numerous cell and gene therapy guidances out there, and if you just Google them, they're on the FDA website, and they range from guidances to sponsors from early stage clinical trials to late stage, and what would constitute the evidentiary standards for establishing effectiveness and safety to approve these products, and how we consider the benefit risk when evaluating the products. There was one mention earlier in the earlier session on extrapolation, which I would like to emphasize since it's a great guidance. It's the ICH E11 guidance, which really culminates the efforts of several regulators and thoughtful scientists, and just to lay the path forward on how to conduct clinical trials and learn from other patient population that are studied, and not necessarily have to conduct studies in every pediatric age group, for example. So it's going to depend on the disease settings. So if the disease that you're considering has similarities between adults and pediatric patient population, if the mechanism of action of the product that you're investigating has expected similar effect in pediatric and adults, there are a lot of data that can be leveraged and extrapolated from either from the adult to pediatrics or even within the pediatric population, studying the several age groups, neonates to 2 to 6, 6 to 12, and then adolescent patients.
Okay. I guess for the two industry folks, when you think about, obviously, the world is changing and rapidly changing, and the availability of genetic testing and such is expansive, right? It's exponentially going. From a pharma perspective, when you decide to treat, and what's the timeline for early symptomatic, pre-symptomatic, in the disease progression, how do you decide what are the factors you begin to weigh and what evidence you do consider in those planning and whatnot?
I think if we think about kind of the clinical development of a new program, we usually will go to understanding, I think, up front, the safety of being paramount and wanting to establish that when we're looking at things for pediatrics. And so our pipeline right now is focusing on Duchenne as our lead clinical indication, our SGT-003 microdystrophin program. We look to kind of establish first a safety, but then to figure out what's going to be the patient population that makes the most sense to be able to also detect a treatment effect. And I think that's where we have to look at things like what are the outcomes that we're able to use within a certain age range, and then how can we potentially be able to move those things into different age groups. So we think about a chronic progressive disease like Duchenne. This is something where you're going to be transitioning through different phases. You're going to be having some patients who are going to be ambulatory, some patients who are going to be non-ambulatory. They can be both symptomatic. You can also have those who are now with, as you mentioned, newborn screening, getting to that stage that could be potentially pre-symptomatic. And I think that's where we want to move in the direction of earlier is likely better. When we think of a disease like this, we're losing muscle over time. And so if we can protect what's there, the impact is likely to be greater. However, I think it's about establishing the mechanism of the drug and getting a good understanding of what does it do to then move in that direction. And I think one of the important things has been, especially for moving into pediatrics in general, is just establishing that prospect for direct benefit and having that to then be able to get your safety, have a potential for that efficacy from your therapeutic, and then potentially get younger and younger. And so most recently in our study, we have actually begun in 4 to 11-year-olds and just opened up a new group that are under 4. So now enrolling those who are 3 years old, 2 years old, 1 year old as well.
Are you then looking at pre-symptomatic conditions then and trying to look at, because the design methodology obviously is very, very different. So how are you dealing with that?
Yeah, so I think as of right now, what we have enrolled have been patients who do have symptoms, early stage symptoms, so at the point of being diagnosed. However, I think it's understanding, can we actually get to the pre-symptomatic, which for us is usually probably going to be in that kind of 1 year range or so. And with that, how can we measure a treatment effect as well? I think that's where we have to think about what are the tools that we have. If we think about something like an ambulatory assessment, those patients will be too young to be able to even show any sort of phenotype in terms of just what the normal standard assessments are. So we move to incorporate things like developmental tests as well. Can we start to see something that establishes a patient's own baseline that then may take longer to be able to see an effect, but you may be able to actually appreciate that over a longer period?
And exactly, I think, thanks, Pat, for highlighting early intervention when it makes sense with safety. I think one of the advantages we had with Zolgensma, which treats spinal muscular atrophy, just very quickly, you may be familiar with it, but spinal muscular atrophy is a monogenetic disease that causes you to lose motor neurons in your spinal cord. So the way the disease works is you have normal intelligence, but you will lose central movement, swallowing, breathing. And It's a very beautiful genotype-phenotype correlation. So in this disease, we did go for pre-symptomatic and develop Zolgensma, which is targeting the root cause. And I think one of the lessons learned here is we saw early treatment, early diagnosis preserves function. And so when you treat pre-symptomatic, you have children that then develop in the normal way. And the other thing that we learned too in doing this is the reason that we were able to get here. The beauty of the story, as Najat mentioned, is the imperative of collaboration. So it started over a decade ago, where really the patient advocacy community, Cure SMA, with scientists, clinicians, regulators, I think the whole ecosystem that's represented here today made a tangible cure possible for monogenetic disease. And I think, Collin, where you were going on the pre-symptomatic is when in cell and gene therapy, if you do have a very strong phenotype-genotype correlation, it brings these possibilities really up to the forefront.
I like that you said working with the communities as well as you're beginning to synthesize and generate the research plan and such. That's interesting and convenient to some of the topics that you were mentioning in the audience in the last session. I mean, I'm going to open this up and whoever feels comfortable, and so I'm going a little off script here based on previous sessions. And Pat, you're ready, but the voice of the patient, we've heard this in a couple of the comments earlier today. I mean, how are you incorporating that, and anyone can speak up to this in the design of gene therapy studies? How are you getting the input and feedback? I mean, this is pretty novel space, risk benefits, really consideration. It's potentially a lifelong therapy, successful or not. And so, that has large implications. And so anyone would like to speak up to that, given the context of what's been discussed, and maybe you mentioned the PFDD meetings and such.
I could take it to start. Yeah, I think one of the most important things is understanding who are the patients that you're treating and what matters to them. What's going to be feasible within a clinical trial, what's going to be meaningful for an outcome that you're measuring. And so with that, I think Duchenne and neuromuscular in general, including SMA, have been two areas where they've been at the forefront of really gene therapy, understanding that this is potentially a single one-time treatment that you're administering. And so the gravity of needing to make sure that you're administering something that could be efficacious as well as safe and also potentially durable, all these factors come into play. As we've done our clinical trial development, we've had actually the great opportunity to work with groups like PPMD. I know some of the Parent Project Muscular Dystrophy folks are here, World Duchenne Organization, others who do put together the ability to really connect directly with the community. And that's been kind of at the core of everything that we've done. So we have collaborations that we do with clinical advisory boards, community advisory boards, patient advisory boards to do protocol review, especially, and take it from the two different pieces I just mentioned of both how can we make sure that we're including the tests that make sense, that are meaningful, and also that are not overly burdensome, that are actually able to be done within the context of a trial. And so both of those have really come together. We have just announced we're going to be running a phase 3 study as well. And so with our ongoing phase 1/2 study, a phase 3 study, each of those protocols, ICFs, have been reviewed by the community for exactly that input.
Okay, Mary.
So I also think this is important for the role of public-private partnerships and my involvement in the International Neonatal Consortium. It has a very multi-stakeholder focus. So it's including the parent voice very actively. And when we talk about cell and gene therapies, the long-term follow-up is a challenge not only for industry, but it's a challenge for families. So in that particular working group, they have the multi-stakeholders, including parents, and they reviewed the guidance documents and looking for similar content and themes. But then they're really discussing what's meaningful to parents and families and what's actually feasible. And they have a paper with their thoughts and recommendations that they're submitting by the end of this year.
That also adds, I guess, a nice transition. But I think one of the elements, particularly in pediatrics, obviously we experience this in oncology, the long-term follow-up requirements in cell and gene-based therapies are really unique, right? They've got a long traction, particularly small and sometimes smaller populations. Do any of the industry members want to speak to how they're experiencing that, what success, any clinical pearls. I think you both are at different stages of that. You may have some lessons learned you'd like to share with the audience and such.
Sure, maybe I'll go first. Is with Zolgensma now, worldwide, we've treated more than 3,000 patients. So this is a rare disease. And so accumulating this type of experience has been really fundamental in our understanding of both durability. So in this group, some of the patients have even been followed up longer than 10 years. And so far, all the evidence we have shows that a single treatment has been durable. So far, the patients are maintaining their milestones. They're not on permanent ventilation. And then in terms of safety, which is also a hypothetical concern with any type of gene therapies. So we look for any evidence of mutagenesis, which is the gene product inserting itself and then posing cancer risk. And so in all of these patients, clinical registries, post-marketing safety, and our industry long-term follow-up, we found no detection of a cancer signal. So I think that's very reassuring in terms of a platform, but also it's going to be very important because now we've given life to children who had a life-limiting disease.
Yeah, and I think as long-term follow-up became a regulatory requirement initially for these sorts of therapies from a safety perspective because of things like the potential for a mutagenesis or insertional mutagenesis, as we're working with viral vectors, that's always been one facet, but I think it's very important to also be looking at that efficacy and at that durability because ultimately the two are going to be important for a long-term therapy like this after a single dose administration, especially in the pediatric setting. And so that's been something that we've always wanted to maintain is as we have a minimum of 5 years of long-term follow-up post-dosing, as is one of the regulatory requirements. We include efficacy assessments at these annual visits as we go out through these periods as well so that we can look to see, are we maintaining changes that we may see early on? Are we seeing anything new that might change over time? And both of those profiles are equally important to characterize.
Najat, do you have any other comments to that about those requirements? Like what types of data do you typically review? Is it registry? Is it, only data from sponsors, follow-up period? What types of things and considerations are that you review for those contexts?
Sure. So during the IND stage and the clinical trial conduct, the data are submitted to us real time. So we do, review these safety reports real time, and we do have a requirement. It depends on the gene therapy and the disease setting. The long-term follow-up can range between 5 to 15 years. After approval of the products, the post-approval requirements for follow-up are mainly for safety to detect, again, the risk of secondary malignancies, any other potential long-term side effects, as well as for to also capture the durability of treatment, since, as was mentioned, the thinking is that these gene therapies are given once, and the hope is that they're going to last for long term. I would just like to maybe just emphasize the point that we all know how burdensome it can be to follow the patients for 15 years, ask them to follow up, and come to clinics and submit test results and so on. However, I think we just have to think in a way where how can we decrease that burden on the patients first, on the sponsors, so that we can collect the data that we need with the least amount of burdens that we can have on the patients. One way is thinking of decentralized clinical trials or just decentralized follow-up for these patients. Can these patients still submit the same amount of data, whether it's a physical exam or blood test? Can they still do it from where they are treated with their primary care physicians without the need to travel to the center where they receive treatment? Can we capture data from registry studies? Can we capture data from real-world data, electronic health records, data claim, and so on? So we just have to think and just be creative and try to see how can we decrease the burden so that we can still collect that information that's very critical. Because when we pre-approval product, our job does not end there as regulators. We need to make sure that the benefit risk is continuing to be monitored and that benefit continues to outweigh the risk of the approved therapies.
I'd love to follow up on that if I could, because I think it's such an important point. And it was mentioned in the last session about kind of how do we find a way to be flexible when we have some of these long-term follow-ups, when we have some of these changes in clinical studies where you may enroll at a specific site, but that is not the most convenient location for you. That's been one thing that's from the clinical development side that we've implemented into our protocols, where we have when we're looking at efficacy specifically, we have a big focus on wanting to maintain our evaluators for a primary endpoint evaluation. And so things like clinical evaluators who have specific training at one site, there might be an importance to be able to maintain that across a baseline to endpoint assessment. However, after that period, we have now allowed for the flexibility to be able to transfer to a site that might be more local to you, one that may not have been open initially that could have come online later, to try to exactly get at this point of collecting the data that's necessary, but doing it in a way that's actually going to be feasible for families, that's not going to be overly burdensome. So we kind of find what that middle ground is. And I think that's something that we're evolving as we come through clinical trial design, especially to be able to incorporate that into designs and then any other long-term follow-up.
Mary, did you want to add anything more from the working group and your experience on looking at either real-world data or long-term follow-up, anything that you like take homes or maybe even describe what you've been looking at?
So the real world data group is conducting a global landscape analysis to identify registries that include children under 24 months of age that have conditions amenable to cell and gene therapies. Well, the good news is that there are data being collected. We assessed 4,000 registries, and about 13% of them do include this population. But when you look, online at what's available about their data practices, it's very limited. So we're in the process now of sending surveys to better characterize their data characteristics so that we can make recommendations about standardization in order to help be ready for the neonates in the infant population to take advantage of cell and gene therapies.
Yeah. Any of your sponsors did you in your planning, what types of data did you use in planning in your study designs? Did you use real world data? Did you use HR data? I mean, what was helpful for you? Obviously, neonates are wild of all us. And Pat or Keely, do you have any comments to that?
I think, the data, it's just so sparse. So any data is so valuable. And I think, with the strategies we have for data now, even an open label study with strategies to try to control for variables. I'm speaking now outside of the Zolgensma study, but within Novartis, we do, like with rarer populations. Currently, I'm involved in a study that has 40 patients within a geographically niche area. And what we're able to do is also dig into our deep databases, look at all the placebo-matched patients that might fit the gender and some comorbidity profile and generate virtual control arms. So I think the future of studying sparse populations with pulling out scientifically robust data is very exciting because there's a lot of technology-enabled solutions.
Pat, did you have anything to add or?
No, I think that's right, especially if I think about other populations, especially when we're thinking about other neuromuscular or other cardiac, I think we have to use what we can to be able to try to work in that direction. No, but I think that's exactly right.
Yeah. We'll switch now a little bit to like sites perspectives, and we talked about decentralized trials a bit, but obviously I remember my site operations days. Operationalize a gene therapy study is very, very different than a small molecule, right? I mean, there's a lot of vagueness and whatnot. And maybe start with Hide, like in Japan, like what types of things have the sites been doing, and what is readiness for gene therapy trials? And then we'll go to industry to talk about what.
Before that, about the long-term follow-up.
Go ahead.
In Japan, well, I talked to my colleague at National Center for Neurology and Psychiatry, and he's heavily involved in the drug development of DMD. And there's a network called Care Cure NMD, and Japan is also a member of a network Treatment NMD. And there's an effort to establish, utilize the already existing registry called Remedy DMD. And it's not good enough for like post-market surveillance. They have to add some more, but they're trying to establish a survey mechanism to cover not only one meds, but as all medicine for DMD. And I think that's a good movement. And there's another discussion among Japanese Society of Child Neurology on certification of sites for pediatric diseases. And I think we'll have a discussion about how to do long-term follow-up maybe next month. So I think there's a lot of discussion about how we can follow up patients in Japan as well. And your question is?
Yeah, more of this, just like you mentioned, you said site certification. What types of things is that going to entail? Is there certain standards that they have to be able to do that are different than normal clinical trials in pediatrics?
Well, I don't see difference, but for example, certification for sites, like for Kimulia, there are 50 sites with accreditation, but accreditation is made by the drug company, not by the Foundation for the Accreditation of Cellular Therapy. I believe that's a third party, but the companies do the accreditation. And the reason why, I guess somebody asked me, like 50 sites is a lot. And I asked the reason to my oncology colleagues, and he said there's a national health insurance that covers gene therapy. That's good. Well, for now, we're not sure how long they can continue to do that. And there are also the Japanese government tried to set up many universities in different prefectures, and they maintain certain quality in medical care, and I think that's good to conduct gene therapies. But I think Japanese hospital has very similar programs in terms of maintenance of quality. Well, our center is like a national center, so we have a very good team, and we have SOPs and manuals for each treatment. But I believe there are the similar problem to maintain quality, like other countries.
Yeah. I think Pat, in our prep, you had other ideas as you were looking at sites and.
Yeah, absolutely. And so I think it's been mentioned a few times so far, but I think the most important thing when we work in AAV-based gene therapies or many more of these viral vector-based gene therapies are going to be the safety evaluations. And so as we look towards establishing new clinical trial sites, we're really looking for the infrastructure to be in place from not just, if we're talking about Duchenne or even one of our cardiac programs, not just the neurologist, not just the primary or the PI, but actually having the support system behind that to be able to operationalize a study. So if I take, for example, the first 2 weeks after a gene therapy administration, you're doing frequent labs. You really have to make sure that you're looking at lab draws for these patients. You're going to be looking at bloods, and then you have to know how to get a good consult for the right thing that you're looking at, and then how to potentially react if there were to be something that would need to have some sort of a reactive treatment. I think that's the most important thing as we look at our core groups of sites, is making sure that, let's say we have a hematologist, a hepatologist, an immunologist, a cardiologist, really in that core group that would be around whoever that primary investigator would be. From there, after that kind of intensive monitoring period that we have for the purpose of safety, now we also have to make sure that each one of the different indications would have the appropriate evaluators that will be there as well. And so again, thinking about neuromuscular indications, these have been things that have been built out for a while from clinical trial standpoints, but we take a lot of time to make sure that our evaluators are trained consistently across sites, and that is kind of that key word, is consistency, especially when you go to different geographies and you're actually going to be looking at different groups that might have different standards of care practices. And so you now want to make sure that that can be as consistent as possible by putting together manuals, by putting together actual training modules that go for each of these evaluators. So I think both of those come in together. And as we're moving into these different indications, like for us, rare cardiac indications, we're moving the neurologist from not a PI perspective, but the cardiologist as a PI and probably a neurologist as a bit of a support system because they have that experience from what they're looking at because they've been there in the past. So it's switching things around a bit, but working from the foundation that we built to be able to make sure that safety and efficacy can be evaluated.
Keely, do you have any thoughts there, obviously?
Sure, definitely. And I think when you're a company like Novartis or any of the big pharma, there's a lot of infrastructure you can leverage. And it's, the decision about being nimble with local versus centralized labs or interpretations. Also data safety monitoring boards, different ways that you can have small groups unblind data when you have like a lot of uncertainty. So I think, Pat, as you mentioned, like all of those things, like on a larger scale, doing multi-center, multinational global trials, like definitely you do need a hub to pull together what your cohesive data signals are and be able to react to those in real time.
Okay. I wanted to, there's a lot of levels of experience in here, and I want to ask one last question of each group before we open it up to questions from the audience. But obviously, you're at different phases of and different lenses of I guess, development, for lack of a word, using a pun on the pediatrics question. But what things would you want people to understand? To me, this is a very complicated space. There's a lot of high expectations. There's a lot of complexity in design. The study designs are quite different from traditional, clinical trials. There's a lot of lack of precedence, et cetera. Are there any thoughts or perspectives that you'd want to share that, either things that you wish you would have known when you started or thoughts that you want to like put out for even others to comment to? Sure, Keely, since you were last, go ahead.
I think be bold because, we are in the era where possibilities, like this is no longer a hypothesis. We can get to tangible cure. I think look for the targets where you have a strong genotype phenotype correlation. And now there are also initiatives that pull newborn screening. I think other countries in the world, when they do newborn screening, they're also doing whole genome with it. So you're getting information that's not necessarily going to be 100% transmitted to the patients and families, but you're getting all this information. So I think, again, it goes back to where we started, the foundation of collaboration. So start with the patient advocacy groups, see where you can combine an unmet need now with a new technology that might bring a possibility of life that previously was unfathomable.
And maybe I can, from, I guess, a regulatory perspective, what I can say is that, as you know, cell and gene therapy products are complex, they're novel. We are learning while the community is learning, we're learning together. I mean, the first chimeric antigen receptor therapy, CAR T cell therapy, was approved in 2017, so not too long ago. So what we're learning right now is that, we have to be flexible. The notion of, as was mentioned earlier today, randomized controlled trial is not going to work for rare pediatric diseases. We have approved products based on single arm trials, relying on the trials being well and adequately controlled, relying on data that support that, whether it's from natural history with really good collection of data that could be helpful in making and supporting those clinical trials, whether it's real world evidence generated data, but to serve as controls. I think, again, like just relying on different paths to move these products forward. And we're open, to hearing what the sponsors propose, as long as they have justification, rationale for why they're proposing certain study design. We work together with the sponsors to make sure that we help these therapies move forward to our patients.
When you're talking about, obviously, this is another conversation that keeps coming at this meeting, is alternative control methods. I mean, are there use cases where you might feel more comfortable with alternative controls, or what type of evidence would support that? Is it, there's a lot of extremes to that. Are there things that you could sort of give us high level, 30,000-foot level considerations of those controls?
I would say, I think the best probably message I would give is that it's a case by case. So if you have any, and probably you hear it a lot from us, and, it is true, because it depends on the disease setting, it depends on the therapy, it depends on even the disease. Is it like early stage? Is it refractory and late stage disease? All of these considerations have to be discussed, and whenever alternative methods are being proposed, as long as there's justification of why they're being proposed and why conventional, randomized controlled study cannot be conducted, or why, preclinical studies cannot move these therapies fast enough, can be discussed.
Yeah, and I think going with that, I think probably the most important thing I would say is for gene therapies, it's not a standard development path. I think there are so many unique features they have to take into account when you're working with not just the modality, but now indications that are going to be for rare disease populations, that are now going to be for pediatric populations specifically. And so I think that's the most important thing, is to be able to understand that there needs to be a level of flexibility. There needs to be a level of working together to understand what will work for this program. I think kind of segueing off of the what's the right comparator to use, it will always be a case-by-case situation. I think even if you think about it theoretically, one comparator is not going to be appropriate for another. But there are many cases where we know that we will have these more rare and ultra-rare scenarios where it's not as feasible to put together a study that might be, well-powered to the extent where we'd really want it to be. But how can we learn still what a treatment effect is? And we have to get to that point somehow. And so I think the most important thing from the sponsor side, industry side, is working with regulators and kind of working through that process together. And couldn't agree more that I think having to do that from an early stage is just of paramount importance. But so I think taking together kind of every unique piece of the journey and just making sure that you're thinking about it up front is really what's necessary.
I have to ask, given you said that, I mean, maybe to you, Pat, since you're on, is like, what was the decision for making you go into pediatric gene therapy studies? I mean, obviously it's given the novelty and, the potential risk, the long-term follow-up. I mean, and you can add to this, Keely, as well. What drove your company to say this is worth it? Because the other thing I think that's been echoing at this meeting is, particularly in rare diseases, is that will people invest in rare diseases? And same thing in neonates. And what was the driving factor for as a sponsor to continue to want to continue that seemingly challenging path and such?
Well, I get to give the good story of how did we start. We started as a company that was focused on Duchenne muscular dystrophy because we were founded by those who were impacted by Duchenne muscular dystrophy. And so with that at our core, it was really that connection to the community, connection to this as being our focus, and wanting to make sure that we're addressing the patients really where they are. And that is really primarily the pediatric patients that we always have to want to expand from there as well. But knowing that that's the point where we could probably come in with a therapy like this. I think when we think about a traditional drug development, you'd likely get that feedback where start in adult and move into pediatrics and move in that direction. For something like a high dose gene therapy that's systemically administered, and you do have to, for Duchenne or for any of these kind of muscle disorders, muscle makes up 40% of your mass. And so that's a lot of vector that you have to get throughout your body if you really have to have that systemic effect. And so that was our motivation to move in the direction of high doses. It actually might be safer in smaller patients. And so that's a piece of the progression of wanting to go younger rather than just, as you might think initially, move into an adult first and then go younger from there. I think when we have these other indications that we're working on, another one that we're newly have announced that we do have our IND clearance and we're working on to go to clinical trials started is for CPVT, which is a rare cardiac indication, catecholaminergic polymorphic ventricular tachycardia. This is something where you're usually talking about pre-symptomatic versus symptomatic. You're pre-symptomatic until you have an event. And that event could be an arrhythmia that actually will end your life at that point. And so many patients are diagnosed once that occurs, if you don't have a family history. And so it's a big motivation to want to go younger and younger, because now you have the ability to actually prevent something like that from occurring. If there's a family history, I think there's more motivation for that. But as things like newborn screening come in and you can identify something like this that has a confirmed genotype phenotype association that would likely cause that over time, I think it makes that more important to work towards moving into that younger and younger population.
Keely, do you have anything to add to that?
Sure, and I think this is, I just caveat, this is probably not the industry-wide approach, but one of the benefits, and I'm really proud to work for Novartis, which is a company that CEO is a physician scientist who spent some time with the WHO. And I think one of the things that we've seen since Voss assumed the role as CEO almost 8 to 9 years now, is he rebranded the company to things really simply: reimagine medicine, bend the arc of life, and be transformational. So we and continue to restructure in the way that we focus on transformational development. And so I think for us, gene therapies just make sense, because where we're going is to meet those criteria. So here I'm going to step a little bit outside of gene therapies, but even in the non-commercial space, Novartis is very committed to malaria. And so we're also working on radical cure for malaria. So I think it's just within our company philosophy to bend the arc of life, which does not mean extend life expectancy, but it really means change the way that people experience health.
So Hide, any pearls for you? Any pearls that your colleagues have shared with you about gene therapy trials in Japan that you would like to share? Obviously, there's a lot of information going on.
I think Japanese neurologists are very keen on international collaboration, and they want to get involved in early discussion. Like when companies come to Japan, and then the protocols are already complete and you can change it. And sometimes, for example, some of the endpoints, some of the biomarkers not validated or something. And to avoid that, it's better to discuss early on protocols, then somehow we can modify the protocol so that it's easier to do in Japan and other Asian countries. And so I think Japanese pediatric neurologists are very in different subspecialties, neurologists and neonatologists. Like there are like more than 10 neonatologists involved in international neonatal consortium activities, and these two groups are very good. So I think having them involved is very good for international drug development.
So Mary, do you have anything?
And I think that we need to address the issue of trust. At least in the United States, research and research institutions are increasingly under attack, and public has been misinformed, and that leads to uncertainty, and people don't know what to believe. So I think that's important when we think of cell and gene therapies, that it's kind of clear that our methods have to be clear, your results need to be reproducible, and your processes need to be transparent. But this is really complex science and information. So I think we need to use all of the health literacy tools available in order to make this information something that people can digest so that we gain their trust.
Definitely. Thank you. We're going to open up the floor for questions for people. Obviously, there are 2 mics. Go ahead. It's not, yeah.
I'd like to follow up, if I can, on the topic that a lot of people were discussing on the specific comparator to use in a drug development program. As many people indicated, of course, this is something that has to be thought about on a case-by-case basis. And of course, it's something that a lot of people in the audience, drug sponsors and advocacy groups, are thinking about a lot. When are natural history data sufficient as a comparator, and when it is necessary to have a placebo control or sham control or some other kind of active comparison group like that. I'd like to ask Dr. Milligan and Dr. Bouchkouj to step us through a specific historic case, and that being the contrast between the approvals of Zolgensma and the approval of Spinraza, which preceded Zolgensma and was approved on the basis of trials which did require sham. I think that the populations, the indications for both those two approvals are very, very similar. The label, and I just look at them to refresh my memory, they both call out the CHOP INTEND as one of the key endpoints. But Spinraza did require that sham design, whereas Zolgensma, the label specifically calls out natural history data. And to my knowledge, those natural history data were not collected under GCP types of conditions and with like Part 11 compliance. Perhaps they were. Can you step us through the contrast between those two approvals?
Maybe, yeah, I'll start and then, so yeah, thank you for this question. It's fascinating. I think this is where it kind of go back in the story of Zolgensma. And I think it came about with a bit of serendipity. I guess like the bus was already in motion generating the data. And I think then it was sort of, I don't want to say retrofitted, but it was sort of already in motion, showing signal. And then it was, found yourself in the situation of what do we do in real time with something that looks very promising, but we can't go back and redesign it to have the comparators that we would have like if we did de novo. And you're right, like it wasn't fully to the standards of GCP and all of that. So it was already a story that was in motion. We saw the signal and we just took it to the finish line with the flexibility negotiation with the regulators. And I think that the commitment from the sponsor side is to make sure that we could trust the efficacy data, that was safe, and that we were generating robust scientific regulatory grade evidence for decision-making that was unconventional.
Thank you for that. While I cannot really comment on specific applications and specific drugs in general, but as I mentioned before, each program is unique. And, while there are general guidances out there, we do have some regulatory flexibility in terms of when a sponsor comes to us and says, this is the clinical study design that I have. And to support that, these are the methods that I'm going to use for control. The natural history, these are the data points that were collected. Were they retrospective? Are they prospectively collected? How confident are we on the data that are being collected that we can count on them because we are making a regulatory decision that we're going to approve a product, and we have to trust that data that are being collected, whether it's a natural history or data collection from real world. All of that, we take all of that into consideration when we decide that a specific control can be used as opposed to another type of control or requiring randomized trial. And in certain instances, certain disease settings, what we've seen is that patients are enrolled in randomized controlled trials. However, later on, there is a crossover, and the patients who received the sham or placebo at the beginning of the trial, they serve as their self-control, and then later on they cross to the receiving the actual therapy. So we are, again, flexible, and although what we need is substantial evidence of effectiveness and safety, and the benefit outweighing the risk when we approve a product. Everything else that comes before can be discussed and negotiated. And that's also, I think, I just want to highlight also that the drug development is a global, in general, like global, development, and we encourage our sponsors to, when they think about the FDA, they think about like other regulatory agencies, and knowing that some regulatory differences exist between the international regulators. We do have at the FDA regular meetings with our counterparts, whether it's EMA, PMDA, Health Canada, Swissmedics, and so on. We do meet with them regularly, especially in products that are really crossing that pivotal trial stage. And at the stage of like really trying to come to market. In the oncology space, we have also discussions with the Oncology Center of Excellence, where we meet also with the regulators to discuss those oncology products. And then in the pediatric space, we have the pediatric clusters. And I have here Lynn Yao and Diana Green from the FDA, who are very critical in these meetings, where we try to align with our international colleagues in terms of studying specific endpoints or coming up with a clinical study design that could be implemented internationally, not just here.
Lynn, did you want to add to that?
Yeah, could I just make an additional comment? Thank you, Najat, and to the whole panel. But specifically to address the question that was raised about the difference between Spinraza and the gene therapy. Yeah. So one of the concepts that you've heard brought up, and we're increasingly using, I think appropriately in pediatric and rare disease development, pediatric rare disease development, is the concept of pediatric extrapolation. And so the more you learn about the disease, the drug pharmacology, the drug, the response to treatment, we are asking people to take that important data and apply it to the next program. So you're not going to see in rare disease, and you're not going to see increasingly in pediatrics, a well-worn development program that you just do the same thing over and over. You are required to take what you learn so that in the next program, you may not need to do as much. Now, I'm not saying that that's exactly the case here, but certainly the concept of extrapolation is that every time you learn something, you should apply that, so that is there something new, different, and potentially less that we need to do to generate the evidence to support approval. So I think that that's a really important concept to remember as we move forward in understanding rare diseases and treatments. You know, the second gene therapy may be different. The second antisense oligonucleotide we may ask for a different efficacy and safety package, again, because we're learning as we go.
Thank you. Ron?
Hi, Ron Bartek. I'm the co-founder and president of the Friedreich's Ataxia Research Alliance. And I want to thank the panel and CPATH for really an excellent discussion of this very important matter. Want to give a little bit of background in that 2.5 years ago, a group of patient advocates and I got together who had suffered the consequences of getting an approval for adults when what we really needed was an approval for children. And so we formed a group that hosted a really powerful discussion on July 16th of last year that was kicked off. It was called the Pediatric Inclusion Roundtable. It was kicked off by the 3 medical product center directors, CDER, CBER, and CDRH, and also by the Neurological Institute director and the NCATS director. They all said the same thing. They said, we have already pivoted. This was Patrizia Cavazzoni in particular and Peter Marks. So we've already pivoted from trying to protect children from clinical trials to trying to protect them with clinical trials so that we give them safe, effective treatments at the best time for them when they need them the most. That roundtable discussion, largely because of the top leadership of the FDA and the NIH saying, we've got your backs. Nobody gets thrown under the bus for doing this, getting these treatments to kids as early as possible, making them part of the same protocol, not waiting 2 years or 3 years or 5 years after adult approval to get the approval for kids. That was so successful that we formed a Pediatric Inclusion Alliance of patient advocates and industry partners. And to socialize that message and to reinforce it from the agency's perspective and to socialize it to our industry partners and patient advocacy group. My question is twofold. One is, how are we doing on that socialization? Are we seeing, for example, in the industry partners here and the agency, are we seeing progress in terms of convincing industry partners that the FDA is receptive to getting children involved in the earliest possible stages of those clinical trials. And the second part is in doing so, we heard early last year that that CBER was forming another roundtable discussion about pediatric inclusion, particularly in cell and gene therapy. That was scheduled for April 4th of this year. Didn't happen. It was postponed and was supposed to happen sometime this fall. So my two-part question is, how are we doing on socialized medicine? That message, do our industry partners believe that the FDA continues to be ready to accommodate early pediatric inclusion? And does the FDA continue to believe that it's ready to accept that? And the second part is, do you have any information on when that additional roundtable on gene therapy, cell and gene therapy for children might take place?
I can start. Okay. So actually, I was the lead for that for the second question that you have for trying to convene a workshop for enrollment of pediatrics in cell and gene therapy trials, in early stage trials, so first in human cell and gene therapy trials. As we all know, things have changed over the last several months. So we are continuing to work towards doing that. And it's going to happen hopefully maybe early 2025 or so. But it is happening. It just, we needed some time to make it happen. And then in terms of your first question, yes. So from our standpoint, again, our thinking as we learn about these therapies has changed over the last several years. We know more about these therapies right now. We know a little bit more about the safety outcomes of these therapies. We definitely, pediatric population is a vulnerable population. We need to protect them. But again, we need to ensure that these therapies, that when they are needed to be enrolled in these therapies, that they are there and enrolled early and not waiting until adults are enrolled first and then pediatrics. So that notion has evolved within CBER, within like our branch for sure. So.
And we have to talk a little bit more later too. So one of our pipeline programs is our FA program, SGT-212. And so I think that's exactly how we wanted to put the program and the study together from the get-go, was with the inclusion of a pediatric cohort as well, understanding that we have a bit of a unique delivery system. We're actually doing both an intradentate nucleus, so actually getting at the ataxia portion, as well as systemic administration. So we're working in that safety evaluation, but progressing in that right direction. So in terms of opening the IND and having a study from the get-go with a cohort that's already included that will be pediatric, no objections were seen from the regulatory side. So we were very happy with that, but we always want to be cognizant of moving in that right direction. And every indication will be a little bit different, but I think this is critical to emphasize for pediatric indications where that earlier could be more important.
I just wanted to add two things, synergy from a policy perspective, which you may already know, but to touch back on Lynn's comment, pediatric extrapolation. So the inclusion of pediatrics doesn't always look like patients in a study. Can you generate the data through modeling and then validation of the model? And so that also counts for pediatric inclusion. And the other policy, which you may know, is in Europe, the European Council for Good Clinical Practice, they have an adolescent inclusion decision tree. So it's an algorithm that helps you, especially for adolescents, when there is enough similarity in their disease, their mechanism, they're not unique vulnerabilities for their developmental age, that they can be seamlessly included into phase 3 trials, or when you would include adolescents. So under that, I don't think it's algorithmic, but at least I think that's progress, and that gives synergy to what we're talking about.
Yeah, great.
So there's a need for strong advocates for pediatric drug development and trying to discuss the culture change that you're talking about. And I have some concerns. So I see trends that I find concerning, such as the disbanding of pediatric centers of excellence within pharmaceutical companies. So that has occurred in the last year and a half. I'm also concerned about some withdrawal of pharmaceutical support to public-private partnerships. We had the Institute of Advanced Clinical Trials for Children here in the US, and that did not achieve sustainability, so that's a concern. We had legislation in terms of pediatric drug development in 2024, which did not advance. So, I mean, I think we need strong advocates to continue to say that we need to protect children through research and we need drug development. And I'd also like to mention that the FDA has an interested parties meeting on the implementation of BPCA and PREA next week, and their public docket is open until September 30th. And so it's really important for patient advocacy groups, clinicians, to be out there sharing the stories of unmet medical needs, and for industry to be saying, you know, what's the right balance of incentives and requirements in order for this model to work within pharmaceutical companies. So I think in the 20 years that I've been involved, that we've made progress, but there is a lot of work to be done, and it has to be done by champions and strong advocates.
Thank you. With that, we'll wrap the panel, and I think people, oh, we have one more. Okay, sorry.
I'll ask one last question. So I'm old enough to remember when we thought gene therapy was going to be easy, right? You know the gene, you know the mutation, you give it and you're done, right? And it's all going to be perfect. And we've all seen that it's way more difficult than that, and that a lot of treatments are modestly efficacious, to be generous. So my question is an ethical one that I think we all think about, whether we're regulators or companies or patient advocates. What do you do when you have, and a lot of these things are AAVs and they're one and done. So what do you do when you have something that is modestly efficacious? You've got desperate patients, you've got desperate parents, but you can't retreat. If it's approved, you can't retreat. So how do you balance how you think about those issues? It's an easy question.
So coming from the perspective of exactly as you mentioned, when gene therapies really came up initially, there was so much of that belief, I think, from the community especially, that this is going to be a miracle. This is going to change from night to day. And I think one of the biggest things is that we need to be communicating effectively. We need to know that there is what the understanding is of a treatment effect, what's the expectation of a treatment effect, so that there is just full awareness of exactly what is going to be administered, especially these contexts when it is a single dose administration. So I think communication is key, working with the community, working with advocates. I think there's always going to be that want for any therapy when there's nothing else that exists. And so I think transparency and making sure that you are listening and actually having a discussion rather than a one-way street is what's most important.
And I'd like to add, I think, very intentional informed consent and also patient selection. So for Zolgensma, just taking that again as an example, there are multiple phenotypes for spinal muscular atrophy, but we went in to treat type 1, so the most severe, very common. And I think that in the development program, as we learn more about Zolgensma, there is efforts to open it up to other less severe treatments. But I think really defining who your responders are going to be very intentionally. And then, as Pat said, communicating transparently on the benefit risk, because it's not risk-free and it's not all benefit.
I think you made an important point, and that I think particularly this question in ultra-rare diseases is extremely important. I mean, you in theory could remove an entire population because they would not be eligible for future clinical trials. And so I think that's a topic that we'll continue to have to encourage, whether it be, a new baseline, I mean, almost treating it almost like the way a clinician would think through if you have an ineffective, treatment. But it's one that will come eventually. We all know that. Okay. I know we're past time and I can feel someone over my shoulder. As far as I'm doing this, she won't leave. No. Well, thank you for the panel.
Thank you. And I'm really sorry. I know there were a couple questions I think that we didn't get to. But we really encourage you, if you have questions for some of the panelists, please find them. Again, just time constraints, apologies, but just a couple quick announcements before we break for lunch. So lunch will be set up out here in the hallway. And please help yourself. We do have 3 breakouts taking place during lunch. So the first one is focused on RDCA DAP task forces. The second one is what happens after approval, rethinking long-term follow-up in pediatric gene therapy. So a lot of the conversation we've just been having. And then the third breakout is implementing new approach methodologies or NAMs in drug development. If you do not remember which session you signed up for, if you downloaded the CVent app, it will tell you there. If you haven't done that, you can check out registration and they can help you. So the idea is to grab your lunch and then go ahead and head to your breakout sessions. Those are up on the 2nd floor in the ballroom, junior ballroom areas. So just take the elevators up to the 2nd floor and there's signage there to direct you. And then I think we have, unfortunately, they are not. Yeah. And then I think Klaus has one more quick announcement.
Let me just do it this way. So, running this type of events with bringing everyone together, and you see it in the passion of the panelists and the discussions and the depth of the conversations that we're having, that takes an army of people behind the scenes making this happen. And I just wanted, before we go to lunch, to acknowledge every single one of them that are standing there in the back that made this happen. So thank you, guys.